Hi, I'm Katie Doroshak. I'm a sixth-year PhD student in CSE, hopefully graduating in the fall. I am part of the Molecular Information Systems Lab, MISSIL, where we're focused on the intersection between biology and computing applied to computer science information problems. I joined the lab in my fourth year, which just goes to show that even in grad school, it's possible to switch gears a little bit to follow your interests. My main project is on molecular tagging, where our goal is to use DNA to identify physical objects. We want to be able to tag an item with DNA, ship it somewhere or store it, and be able to identify it at a later date. Some applications of this include tracking and provenance, secret exchange, and counterfeit detection. You can think of this as a biological QR code or RFID tag, only it can be used in situations where maybe the product is too wet or numerous to use existing technology. We use nanopore sequencing to detect the molecular tags. Some of the requirements for this system is that it needs to be DIY end-to-end by non-experts, including computer scientists like myself. We want to be able to generate arbitrary tags on demand without doing DNA synthesis. DNA synthesis is a process where you essentially send a company a DNA sequence, they will make it for you and ship it back. This process is very expensive and can take a long time. We want to be able to decode quickly and accurately and use minimal special equipment. And for this, we use a nanopore sequencing device, which is pictured in this slide. And this thing is about the size of a Snickers bar. It plugs into your computer using just an HDMI cable and is super convenient, especially compared to benchtop sequencing machines that are about the size of a big printer. Nanopore sequencing works by unwinding DNA through a tiny pore while measuring ionic current as it passes through. The current changes depending on what DNA is in the pore, so we can convert the signal back into a DNA sequence if we want to. We call this system Porcupine, and here's how it works. We start out with a digital tag made of ones and zeros. Each bit in this tag is assigned a different type of DNA and is combined so that if a bit is set to 1, it is physically pipetted into the tag mixture. If it is 0, it is left out. We call these different types of DNA molecular bits, or mole bits, and the final product is a molecular tag. Then we apply this mixture to an object, ship it or store it, and rehydrate it when we're ready to read it. We then load it into the MinION and identify the tag using software. Our goal then becomes trying to design these DNA strands so that they are easy to distinguish. Here's the DNA design that we came up with for a single molecular bit. Each mole bit is made of a unique sequence and a specific length. We will have lots of combinations of unique sequences and different lengths in order to produce a full set of mole bits. So why only use part of the strand? One reason is so that we can combine more bits without synthesizing more DNA. By adding just one more length to combine with all of these unique sequences, we double the number of available bits, and it's really easy to distinguish lengths on the nanopore. Another reason, and the one that's way more interesting to me, is that this unique identifiable region means that we can avoid reading DNA directly. We're essentially turning a really complex decoding problem called base calling into a classification problem. This is fundamentally an easier problem to solve, and accuracy can be much higher. So, to make this classification easier, we designed molecular bits to produce unique current squiggles. We used a tool to take a DNA sequence and predict what the nanopore signal should look like. Here we simulated a DNA sequence that produces the letters UW, and it's actually pretty accurate. This means we're able to use this to help design strands to look visually different and therefore be easier to classify. Going back to that diagram of what the DNA design looks like, here's where the unique barcodes go in the strand. We'll then throw these raw signals as they're produced by the nanopore sequencing device into a convolutional neural network to predict which mole bit is present. There's a little bit more to the decoding that I can get into in a lightning talk, but essentially we'll observe many of these strands and count how many of each mole bit is present. Then we need to know two things: how much time it takes to decode and how reliable it is. This is a dense plot, but we'll break it down. On the x-axis, we have the sequencing runtime, how long we ran the nanopore. It's on a log scale over seconds. The y-axis is the number of bits that were incorrect when decoding. We have some error correction built in so that anything under 9 errors is guaranteed to be correct or correctable, and that's this dashed line. We might be able to get it correct when we have even more errors, but it's not a guarantee. Then each x on this plot is one incorrect decoding. When testing multiple sets of tags, we get the answer right every time after only 10 seconds of data, which is pretty awesome. With that, I'd like to thank you so much for watching and for attending this event, even in these crazy times. And I'd love to thank the MISSIL Lab, who I've truly loved working with. This QR code that I have here goes to the preprint of our paper in case you're interested in reading it. And feel free to reach out to me if you have any questions or want to chat. Thank you.